Today we're going to go over enzymes, which are proteins that have catalyst function in biological systems. Before we start talking about enzymes specifically, we're going to review a couple vocabulary terms. So a chemical reaction is when reactants interact with each other and produce a new product. So the reactants are no longer there at the end of the reaction because they've changed the atoms around to create a new molecule. A catalyst lowers the activation energy needed to start a reaction, and so it helps reactions to happen more quickly. There are two major types of chemical reactions. There's endothermic reactions, and they absorb energy. And then there's also exothermic reactions, and they release energy. So when a reaction absorbs energy, it often feels cool to the touch while it's happening. Exothermic reactions generally release energy, and some of that is as heat. So exothermic reactions are things like fires, for example, that give off heat as energy. And you can see in these graphs the difference between the two. So an exothermic or exergonic reaction has a energy output in the products that is lower than what we started with. And because endothermic reactions absorb energy, the products have more energy stored than the reactants did. An enzyme is a protein, that's important. Enzymes are made of proteins, and their job is to speed up chemical reactions. They are known as organic catalysts. So they speed up reactions like a catalyst, but they're organic because they're made of protein. Enzymes are also reusable, so once they've facilitated one reaction, they can move on to the next. And they kind of act like a lock and a key. They make reactions happen. Part of that is that they lower the activation energy needed to get a reaction started. Activation energy is just the energy needed to start a reaction. So anytime a reaction happens, usually you'll have the reactants, and they're going to be near each other, but they might not be reacting with each other. If you put in a little bit of energy, so that's this, that allows the energy or the reaction to start. And then in this graph, for example, that reaction is exothermic because there's less energy stored in the products than there is in the reactants. But this activation energy is required to help the reaction get going. If you use an enzyme in a chemical reaction, that activation energy decreases. So the top line here shows a reaction happening without an enzyme helping the reaction happen. And you'll notice in the second line here, with an enzyme, that the activation energy has decreased. So it takes less energy to get the reaction to go if an enzyme is present. And that helps chemical reactions happen more quickly. When an enzyme is working, the molecules that are reacting are called the substrate. So in this image, you can see this is the enzyme. And the enzyme has this place called the active site. And that's where the substrate can bind. And so the substrate fits into the active site, like you can see in this image. When the substrate binds to the enzyme holds it in place so that the reaction can happen more quickly. And in this example, as the reaction is holding it in place, the reaction is to break two things apart. And so at the end, you'll notice that the enzyme itself has not changed, but now we have two products when we started with only one reactant. So like we just talked about, the reactant is the molecules that we started with. So in this example, the reactants are hydrogen and oxygen, and notice that they're not bonded to each other in this example. Then a chemical reaction happens and it makes the product, and now you can see that the oxygen is bonded to two hydrogens and we have made water. So the reactants are what you start with, and the product is what you end with in a chemical reaction. So the product is what you make. So again, the product is what we have produced, hence the name. And so products, again, are what we make in a chemical reaction. Reviewing again, reactants are what we start with. enzymes are very specific to what substrates they will work with. So they're kind of like a key. The key to your house is probably not going to start your car, for example. You have two separate keys for two separate activities, right? The key for your house unlocks your house. The key for your car starts your car. And so they're site specific. So that means that one chemical reaction can be facilitated by one enzyme, but that same enzyme will not do a different reaction. So like in your digestive system, for example, there's an enzyme called amylase, and it breaks down starch. It only breaks down starch. If you were to eat a piece of chicken, the amylase enzyme will not break down the protein in the chicken you ate. Enzymes can work in a couple different ways. The first way is called degradation, and that means it breaks things apart. So when you have a degradation enzyme, the substrate binds to the active site, and then it's broken into two or more products. So that's breaking things apart. The opposite of that is a synthesis reaction, and that occurs when two substrates or more bind to the active site, and then they're bonded together to make one product. So synthesis enzymes join things together. Degradation enzymes break things apart. You usually know that a molecule is an enzyme because of how the word is written. So enzymes end in the suffix ase. So lactose is the sugar, and the enzyme lactase breaks it down. Amylose is a sugar. Amylase breaks it down. Lipids are fats, and they can be broken down by lipase. And hydrogen peroxide is broken down by peroxidase. So that suffix is often a key that you're talking about an enzyme. Because enzymes are made of protein, they can be damaged if they are in the wrong environment. So there's a few things that can slow enzymes down because it changes the shape of the enzyme. One of those things is pH. If the pH is incorrect, that does something called denaturing the protein or denaturing the enzyme. When something denatures, it means it changes the shape. So if you were to change the shape of the key to your house, it wouldn't unlock your house anymore. And the same thing happens with enzymes. If you change the shape of the protein by changing the pH, it can no longer carry out its job as a catalyst. Another thing that can slow enzymes down is temperature. So enzymes like to be at a specific temperature, and if that temperature changes, it changes the shape of the enzyme and it's no longer functional. So here you can see a couple of graphs that demonstrate the effective range for some different enzymes. So this enzyme you can see is most reactive at a specific temperature range, around 35 to 45 degrees or so, or 50 degrees, somewhere in here. So this is the temperature range where this enzyme likes to work. If it gets too cold or too hot, you see that the enzyme is a lot less productive because the rate of reaction has decreased. The same thing can be said for enzymes and pH. So in your body, you've got a couple different enzymes that break down proteins. One of them is called pepsin. Pepsin works in your stomach, so you'll notice that it works best at a pH that's quite low, around 2, because your stomach acid is very acidic. That's different than trypsin, which works in a more alkaline environment, and that is in your intestines. And so pepsin does not work very well in your intestine. Trypsin does not work very well in your stomach because it's too acidic. So like we talked about before, when something denatures, that sensitivity to pH, for example, changes the shape of the active site. So normally, if the pH is correct for this enzyme, the substrate in this example is shaped like a square, and the active site is also shaped like a square. If the pH is changed from the optimal pH, then even though the substrate's still the same shape, the active site has changed because the protein denatured, and that makes this protein unable to act as an enzyme, and so the reaction gets a lot slower. Temperature has the same issue. At an optimal temperature, so in this example, something below the 40-degree mark, you'll notice that the substrate and the enzyme will fit together. If it gets too hot, the enzyme changes shape. You'll notice the active site specifically has changed shape So it will no longer bind to the substrate. That means it has been denatured and is no longer very functional. You can also slow enzymes down if the available substrate is not available. So like if there's very little reactants, then the enzyme is going to be slow. And if you don't have enough enzyme, that can also change things. And so there's several different things that can impact the rate of reaction with enzymes.